Hello friends, welcome to Nanoscience Technology. This is Mike Escuti. I want to talk about near-field imaging and near-field light. And this may sound like a strange set of words, but I hope you can, after this lecture, you can understand them with a bit of confidence. Now, near-field light has to do with the light that is close to a surface within a wavelength. And that's very different from conventional imaging systems. So let's first write down what a conventional imaging system is. And of course, this includes most cameras and microscopes that you are familiar with. Now, the most important thing about a conventional imaging system is that they have a fundamental resolution limit. And this goes by many names, but really it's the same thing. And one name for this is the Rayleigh criterion. This is also called the diffraction limit. Among other names. And what this tells us is that there is a minimum resolvable feature size. And this is a real spatial dimension, so it's in the scale of nanometers or microns, and it can be calculated by this expression, which we're not going to derive because it's besides the point here, but it has to do with the wavelength, as you can see, some constants, the index n, or the refractive index n, and sine of an angle. All right, so let's identify n is the refractive index. And this is the refractive index of the medium through which the imaging is happening. In other words, if you are taking a picture with a smartphone of a scene, then the medium, of course, is air. If you're taking a scene of for example, a selfie, right? So then, in that case, the medium is air. If you are underwater, then the medium is water, and it has a different index of refraction. Or if you're in a semiconductor photolithography setting, there may be an oil with yet a different index. And what this means is that this minimum resolvable feature size is that if you have two features next to each other spatially, let me try that again. If you have two features that are spatially next to each other where the physical structure may be something like this, whereas the imaged feature may look something like this. It's not going to be as sharp eventually, and they could be close, right? So this is a kind of distance that we can identify between these two. And what we're saying is that there's a minimum resolvable feature size when this structure gets so small such that the imaged structure looks something like this. And so this looks like a single feature, but really it's two, right? So in this case, this would be a distance that is smaller than the criterion shown up here, whereas this would be larger than the criterion. All right, so that's the basic idea. And the central point is that this limits conventional imaging to something around a couple hundred nanometers, which is about the wavelength of light. So really high-end microscopes might get down to 200 or 300 nanometers, whereas normal everyday microscopes that would be found in most labs might be limited to about an order of magnitude larger than this, a couple microns. All right, and this is all called far-field imaging. Because lenses are used to take the image of the surface or of the scene and in a way relay it onto a detector that is far away from the surface. All right, so let's contrast that with near-field imaging. So I'm just going to outline it quickly here, and then I'll let you watch another better produced video about near-field imaging. So this is a scheme for the detection of photons that are emitted from a surface or that are reflected from a surface or through a surface, where the detection happens before they travel away or very far away from the surface. And with this principle, we can get around this diffraction limit, or the Rayleigh criterion. And of course, in this case, there must be some metric of what is close to the surface, what is far away. And generally, this means that near-field imaging happens within one wavelength of the surface. All right, so it's important in this imaging scheme to have a detector that's very close to the surface, or a source that is very close to the surface. Now, before I show you the actual microscope, I'd like to see if I can convince you of what the principle is here, and that actually it makes some sense. Now, this uses evanescent fields, and although that sounds like a really complex term, or maybe mysterious, it's really not. And I'll draw you a picture which I think can give you a sense of why this works. All right, and an evanescent field decays exponentially. And because it decays very rapidly, exponentially, that's why it's important to either have the source or the detector very close to the surface. And this method can get down to perhaps 20 or even more commonly 50 nanometers up to, let's say, 200 nanometers. So that means we can optically resolve something in the tens of nanometers, even though that's about a tenth of the wavelength of light being used. All right, so how can this possibly be? Well, let's consider an island, a bean-shaped island, something like this. Or I suppose you can think of it as a pebble. Either way, same thing. Now, if we were going to drop that pebble into a tank of water, or if we were looking at an island from above and looking at how the waves impinge upon it, we would expect to see that there would be a series of waves, something like this, around this island. And the key thing I want to emphasize here is that very close to the island, the waves, for example around here, would follow the shape of the island very closely. However, for waves that are farther away, that would get averaged out, and the feature of the island could not be seen by these waves. And then, of course, the lesson is that this would be the far-field wave, and this would be normal conventional imaging, whereas over here this would be near field. All right, so that's the basic principle. We need to get a detector or a source very close to the surface. And the typical setup All right, now the typical setup has two parts. First of all, a sophisticated source, which is an optical fiber that's metal coated and tapered. And then secondly, a dumb detector, which is simply a photodiode or something detecting the light itself. So let's draw what this looks like, and you get a sense of how this microscope works. Now, let's begin by drawing the sample surface. And let's say there's some nano pattern on the surface here that we want to image. Now, in this case, we have, the idea is to make sure that we have a fiber optic probe. So this would be the probe. And this would then be scanned across the surface back and forth, while at the same time the light that comes through and out through here would be detected by some kind of detector, a photodiode. And the key thing is that the size of this bundle of light emitting from the edge of this could be about 20 to 50 nanometers. Right, so this is the sample. And most importantly, the distance between the edge of the probe and the surface or the sample should be less than the wavelength, right? So that's the key for this to work. All right, so in many ways this is very, very simple. However, as I hope you can imagine, there are some really important disadvantages as compared to a conventional microscope. What are they? What do you think they might be? Well, first of all, it's very slow, because the probe tip has to scan the sample spatially, and it has to do it at a really tiny resolution. Secondly, the probe must nearly touch the surface. And as you can imagine, that's very hard to control, especially for a sample that has a varying surface. And so there's lots of techniques on how to do that. And then lastly, there's a very small depth of focus. And so practical near-field optical microscopes have a whole lot of technology into controlling this distance and sensing and detecting how far away the probe should be so that they can get reasonable resolution. All right, so this is near-field optical microscopy, and it's based on this idea that the probe emits from a very small aperture that then is spread out into the sample, and that's detected by usually a basic dumb detector. But the benefit is, of course, that the microscope can resolve things at much, much less than a wavelength of light.